Can you talk about risk and gene expression profiling?
There's other ways in which we can talk about risk, and one of them is this gene expression profiling, and you probably will hear a bit more from Dr. Morgan, which can, it's another way to look at prognosis and try to separate those patients that actually may have a more aggressive form of myeloma. So that's one of the ways. There's actually a commercial assay that's available too for doing this through this Sky company that can test for the high risk markers. Now, one of the things to think about with these two things is that, again, number one, prognosis is not determinant. It's not a black and white. You have this, you're going to do well. You have this, you're going to do bad. We still are going to work together through those things. And the other thing is that these are only approximations because there's many other things that come into play. If you come to the clinic and a person has severe diabetes and heart failure, it's going to be a lot harder to get going through the completion of the treatment. So all of that plays out on how we're able to propose different treatments for patients.
What is gene expression profiling, or GEP?
Gene expression profiling turns out to be a really useful tool. It's quite hard to do, but I think it's now commercially available. Gene expression profiling doesn't look at the protein, it looks at that RNA that takes the information from the DNA to the protein. And you can use it to do a variety of things. You can use it to classify myeloma into 6 broad groups. It makes it very easy to do that. But what it was classically used for was to make prognostic scores. And there's one called the GEP 70, which is basically a 70 gene score that predicts 15% of people with myeloma who really don't do well with current treatments that are or should be the subject of clinical trials. We should be doing those trials which are called risk stratified trials to really try and improve the outcome for that group, because the reality is in the last 20 years their outcome really hasn't improved hardly at all. In contrast, the low risk patients are now doing really well. So there are other tests. There's one called the EMC 92, which is a very similar test. The problem has been accessing those tests, and so only some centers have taken the trouble to establish them, and so most people don't have that information. I think it will change with these DNA panels because they're easier, they're more robust, people are investing in the technology, and so I'm hopeful that that's going to be the way that we risk stratify patients in the future.
Can you explain what a gene expression profile is?
One way to look at that is also, instead of just looking at the DNA code, is to look at these RNA molecules. These are the molecules that are expressed from the genes. So the genes express the RNA then is translated into the protein. The more RNA you make, the more protein you get. If we can try to understand the characteristics of tumor cells and how much of a gene is being expressed, that could be useful information too. And what I'm showing here, what's called a heat map. Let me explain that. We now have the capability of looking at how every gene in the cell is being expressed. And we have about 17,000 genes in our genome. So there's about 17,000 genes that give us all the functions we need to be a human being. Different genes are expressed in different cells. So a muscle cell will express genes, a liver cell, a plasma cell, they'll express their sets of genes. But what we really wanted to understand was, how does a normal plasma cell express genes compared to a myeloma plasma cell? They're both plasma cells. You would expect them to have certain genetic characteristics in common, but the genes that they express may be different in the tumor cell than in the normal plasma cell. And the heat map that I'm showing here demonstrates every gene that I'm looking at is a row. So a different gene, each row is a different gene. Every column is a different sample. And you'll notice on the left side, there's a label there that says NPC. Those are normal plasma cells. And you'll notice there's a set of genes that are red. That means they're expressed at high level. There's a set of genes that are green. They're expressed at a very low level. But what we really want to do is compare that then to the myeloma gene expression pattern for those same genes. And look what happens in myeloma. All those genes that are red in normal plasma cells aren't expressed very well in myeloma. All those genes that are not expressed much at all in normal plasma cells are now highly expressed in myeloma. So we're understanding a pattern here. There's a pattern of genes that are changing in their expression that distinguish a myeloma plasma cell from a normal plasma cell. And that tells us something about the biology of this disease. Now look at those myelomas and the columns, and you'll notice that even though there's a block of red and a block of green, those two columns are not identical in every myeloma. Brings home my point. Every myeloma is different. So even though there are some common genes that are often upregulated or genes that are downregulated in their expression, every myeloma is a little different. And really, we need to understand that. In fact, you could take that pattern there and you could say, I've got a pattern. Where else do we see patterns that distinguish outcomes? One of those is in a grocery store barcode. So I'm giving you an example here of a pattern. These are two grocery store barcodes. They have some things in common, some things that distinguish them. It's a barcode. Look at the bottom of the barcode. You'll notice that the bars all look very similar. That means those two products in the grocery store probably share something in common. But look, as you move up the pattern starts shifting, which says, wait a minute, those two barcodes probably are defining two different products in the grocery store, but something that they have in common because the pattern is similar. Turns out one of those the scans says, well, this is wheat bread, and one of them is white bread. They share a common feature. They're both bread, but they are distinct. One is white, one is wheat. Let's think about that application of a barcode that might distinguish a patient who will respond to a therapy versus a patient that doesn't respond to a therapy. And can we, in fact, use a scanner to scan a pattern of gene expression that will distinguish a response and a non-response for a particular therapy? And that's work that we've done and other labs have done as well. So here's an example of a tumor, one myeloma. This myeloma was being treated with bortezomib. And this is just a laboratory treatment of the myeloma. And you can see in this graph, what you're seeing is 100% of the cells are alive when we started, but we started giving this tumor in the lab bortezomib or Velcade, which a lot of you are familiar with. And you'll notice that at 100%, as we increased the concentration of the drug, we had fewer and fewer cells surviving, so that by the time we got to a relatively low concentration of drug, we killed all the myeloma cells. That would tell me that that myeloma right there had a pretty good response to bortezomib because we killed all the cells. Now the question is, if I test other myelomas, what happens? Well, what you can see here is I've got a lot of myelomas that look like that first one where they die very quickly in the drug. But look at all the myelomas that aren't responding very well. That's a demonstration of the heterogeneity of myeloma from different patients. Some of those patients' myelomas responded, killing all those tumor cells to bortezomib very quickly, but there are a lot of myelomas that didn't respond well to that at all. How can I tell the difference? What if I could generate a barcode of a heat map of a gene expression that might distinguish a responder from a non-responder? So what we did is we collected on the left side all the guys that responded, and on the right side of this graph all the myelomas that didn't respond. And then we put them in these groups and said, let's look at the gene expression pattern of responders and non-responders and ask the computer if it can identify a set of genes that will distinguish those two. And here is a result. I now have another heat map. But my heat map is not distinguishing normal myeloma from normal plasma cells from myeloma plasma cells. The pattern on the left are all the myelomas that responded to the drug. The pattern on the right are all the myelomas that did not respond to the drug. That could be useful information. So for example, if a new patient walks in the door and I say, you know what, before we even treat you, let's do an expression analysis of your tumor. And if we generate a heat map of one tumor and it looks like that, you say, gee, that pattern matches the ones on the left that are responders. This will be a good drug for you to try. If your pattern looked like the one on the right, which were all non-responders, you might say, you know, based on your gene expression pattern, you might not respond to the drug. Maybe we should find other drugs that you will respond to.
So gene expression profiling is a way of looking at all the messenger RNA that a myeloma tumor makes. It can be done in many different ways. Sort of the traditional way has been something called microarray, which is one genomic test for looking at it. A more modern way would be through next generation sequencing. But either way you do it, you still get the gene expression profile, which is a list of all the genes in the cell and how much of each one there is. And that has been done, and it's been shown that it really serves to divide up myeloma patients into different groups, which correspond pretty closely to the fish, in fact. But it gives you more information than the fish. So it doesn't only tell you about the chromosome translocation or maybe gains or losses of chromosomes, but it really tells you about the behavior of the cell. Is it making a lot of genes associated with cell division, which would be something we'd associate with a poor prognosis. And in fact, you can get much more sophisticated than that and develop prognostic indices that can be used to say, okay, this patient has got high-risk disease because the tumor has the expression of all these different genes. There are two tests, the Sky92 and the MyPRS, but there have been issues with insurance reimbursement, and I believe the MyPRS is no longer available. I do not believe that the Sky92 is widely used, but it is a useful test and can tell you a lot of information about the tumor.
How is gene expression profiling, or GEP, done?
Gene expression profiling can be done with what's called microarray, and that's the test like the MyPRS or the Sky92, and it's sort of somewhat older, like 10 years old. Whereas most people now who are doing gene expression, at least for research, would use sequencing, which is a more robust technique. I don't think there are any commercially available sequencing tests for gene expression in myeloma. The way it works with gene expression profiling in myeloma is that we actually take a genomic approach, meaning we look at all the genes. And we don't just look at a select few. Mind you, of all those genes, there's typically only a few of them that are really interesting, but we still look at all of the genes. But then, for instance, for the Sky92 test, they focused in on just 92 out of the 30,000 to make the score that they used to determine prognosis. And it was a similar number for the MyPRS test. Often you'll see gene expression profiling summarized with a heat map with green and red, sometimes blue and yellow. The green often is low, the red is high, same with blue and yellow. And what you're doing in that case typically is you're taking just myeloma patients, so you don't have normal cells in there, you don't have other tumors, you just have myeloma patients. And you're looking within a group of myeloma patients, how do the genes differ in their expression? And you'll find that certain patients have a very characteristic profile. They all express the same kind of genes. And that's how we group patients into different biologic types. And those biologic types often correspond with the genetics. So, a classic example would be the 11-14 chromosome translocation, where there's a gene on chromosome 11 called cyclin D1 that gets turned on by that translocation. And so when you look at the gene expression profiling, you see that in those group of patients, there's sky-high expression of cyclin D1.
Can GEP help guide which treatments to use?
That's sort of the holy grail of sort of genomic testing, is to identify models that will tell us which patients will respond to which drug. And there's very provocative data from Dr. Chapman in the United Kingdom, who identified a profile, and if, depending on how patients scored, they were either likely to respond to proteasome inhibitors like Velcade or Kyprolis, or they were likely to respond to IMiDs like Revlimid or thalidomide. And that signature that he used, I think, was like 6 genes, a very small number of genes. And so he's, I think, looking to validate that in another set of patients to see if it's real. And then if it is, it could be a useful tool. But currently, there isn't anything that we can, we don't have a practical test right now.